process that all living organisms undergo to copy their DNA. It is an elaborate process that requires several enzymes such as helicases, topoisomerases, DNA polymerase, and DNA ligase to catalyze the replication reaction. In a cell, DNA replication begins at a specific point. For example, bacteria E. coli has a definite region where the DNA replication originates. Such a region is known as the origin of replication. This is also the reason why a vector is required when a piece of DNA needs to be replicated during recombinant DNA procedures, as it is the vector that provides the origin of replication. In some cases, like eukaryotes with large DNA molecules, there may be many origins of replication that finally merge with one another. During replication, first the enzyme helicases unwind and uncoil the DNA double helix into single strands of DNA by the breakdown of hydrogen bonds. However, the main enzyme that catalyzes the replication process after it begins at the origin of replication is DNA polymerase, which is said to be DNA dependent as it uses the DNA template to catalyze the polymerization of deoxyribonucleotides. The DNA replication process requires a high degree of accuracy because any error during replication will result in mutations. Thankfully, DNA polymerase is a highly efficient enzyme that catalyzes the reaction not only with accuracy, but also does it very swiftly. It catalyzes the polymerization of a large number of nucleotides in a very short span. For instance, E. coli can replicate 4.6 into 10 to the power 6 base pairs of diploid content in 38 minutes. That is, the average rate of polymerization is 2000 base pairs per second. The replication process is also an expensive process in the sense that it requires expenditure of energy. The deoxyribonucleoside triphosphates that act as substrates also serve as the energy source for the reaction. The terminal phosphates in the deoxyribonucleoside triphosphates break down to provide energy as ATP. Due to the high energy requirement, two DNA strands do not always separate completely, especially in the case of long DNA molecules. However, to facilitate the separation of a DNA helix, the enzyme topoisomerase cuts and rejoins one strand of DNA. This then unzips the double-stranded DNA. As a result, replication bubbles are formed that extend as a Y-shaped replication fork. This replication fork is actually a small opening in the DNA helix where replication occurs. However, there is a small catch. The DNA-dependent DNA polymerase enzyme can catalyze the replication reaction in only one direction, 5' to 3'. This causes additional complications. Replication on one template strand that has 3' to 5' polarity, also known as the leading strand, is continuous, whereas replication on the other template strand with 5' to 3', also referred to as the lagging strand, is discontinuous. These discontinuously replicated fragments, also known as Okazaki fragments, are then joined by the enzyme DNA ligase. Further, cellular proofreading mechanisms ensure the fidelity of DNA replication. In case wrong bases are entered into the helix of DNA by mutation, the enzyme nuclease can identify and correct them. They cut off the defective segment of DNA and introduce the correct segment, which is then joined by the enzyme DNA ligase. However, scientists are still to understand all the aspects of DNA replication. Although it is a known fact that replication of DNA in eukaryotes takes place in the synthesis phase of the cell cycle. The cell cycle too needs to be coordinated with the replication process, or a chromosome anomaly may occur. For example, a failure in cell division after DNA replication results in an inheritance disorder as polyploidy. Therefore, the process of DNA replication, which is the basis of biological inheritance, is conducted with accuracy with the help of various enzymes in a determined manner. Proteins play a pivotal role in the proper functioning of any organism. The synthesis of proteins occurs in the cell in two phases, namely transcription and translation. Transcription takes place in the nucleus, where the DNA acts as a template for the synthesis of messenger RNA. It is a process by which genetic information in DNA is transferred to a molecule of messenger RNA. It is governed by the principle of complementarity, with one exception, that adenine now forms base pair with uracil instead of thymine. Messenger RNA carries the genetic information to the cytoplasm for protein synthesis, and hence it gets the name messenger. The transcription process is different from DNA replication. In replication, the total DNA of an organism gets duplicated, while in transcription, only one DNA strand gets copied into RNA. In case both DNA strands act as templates in transcription, two types of proteins will be produced. One with the correct sequence and the other with the reverse sequence of amino acids. Moreover, the simultaneously produced RNA molecules would become complementary to each other and will form double-stranded RNA. As a result, RNA will not get translated into protein and the process of transcription will get nullified. However, such an occurrence is quite rare. The segment of DNA which takes part in the transcription process is called the transcription unit, and it encodes for at least one gene. Let's understand the transcription unit in detail. It consists of three main regions, namely the promoter, the structural gene, and the terminator. The promoter is the starting point of the transcription process and is present upstream at the 5' end of the structural gene. In the promoter, there is a binding site for RNA polymerase, which moves along the DNA strand from 3' to 5' direction. With the help of RNA polymerase, the promoter can code for the two strands of the DNA helix. However, these two DNA strands will get reversed if the promoter switches its position with the terminator. The next part of the transcription unit is the structural gene, which consists of two DNA strands having opposite polarity. The strand with the 3' to 5' polarity acts as a template and is called the template strand. while the DNA strand, which has 5' to 3' polarity, is known as the coding strand and is displaced during transcription. The sequence of this strand is the same as that of RNA, except that thymine exists in place of uracil. The structural gene can be defined in terms of a cistron or segment of DNA that codes for a polypeptide. The structural gene in a transcription unit can be either monocistronic or polycistronic. A monocistronic structural gene contains the genetic information to translate only a single protein and is found in eukaryotes. On the other hand, a polycistronic structural gene carries the information of several genes which can be translated into several proteins. It is found in prokaryotes. The monocistronic structural gene has a split gene arrangement, where sequences called introns and exons are present. However, in this gene arrangement, only the exons act as coding sequences. The process of transcription helps remove introns and joins exons as functional messenger RNA. It is the exons in the structural gene, along with the promoter, which determine the inheritance of a character. The structural gene also consists of regulatory sequences which regulate the functions of other genes but do not code for any RNA or protein. They are also known as regulatory genes. At the 3' end of the structural gene is the terminator, which is another important part of the transcription unit. The terminator defines the end of the process of transcription. Transcription also happens to be the first stage in the process of gene expression. As it helps in the transcription of a gene into messenger RNA and encodes for a protein. The building of protein is very essential to the functioning of an organism. In this protein building task, DNA acts as a master blueprint containing all genetic information about the protein to be created. The genetic information from DNA is transferred to messenger RNA or mRNA through a process known as transcription. Now let's learn how transcription takes place in prokaryotes such as bacteria. The prokaryotic cell does not have separations in terms of cytoplasm and nucleus. Therefore, both transcription and translation processes involved in protein synthesis can take place inside the prokaryotic cell at the same time. In prokaryotes such as bacteria, only one DNA-dependent RNA polymerase enzyme catalyzes the transcription of all types of RNA. There are 3 types of RNAs involved in the process of protein synthesis: messenger RNA or mRNA, transfer RNA or tRNA, and ribosomal RNA or rRNA. The mRNA carries the coding information to the sites of protein synthesis. It helps to put together amino acids to make protein. The tRNA carries each amino acid to the ribosome according to the coded message in the mRNA, while the rRNA provides a mechanism to decode mRNA into amino acids. Transcription The first process in protein synthesis occurs in 3 stages: initiation, chain elongation, and termination. In the first stage, RNA polymerase, along with the initiation factor denoted as sigma, binds to the promoter sequence in DNA and initiates transcription. This helps in the opening of the DNA helix and separates the 2 DNA strands. Now, the second stage of transcription, called elongation, begins. In this stage, the RNA polymerase builds a strand of RNA using one of the DNA strands as a template. RNA polymerase uses nucleoside triphosphates as substrates and polymerizes using the law of complementarity. This RNA chain growth takes place in 5' to 3' direction. After chain elongation commences, the sigma factor dissociates from the RNA polymerase and can be reused. The final stage of transcription is termination, in which the polymerase, along with termination factor represented by Rho, reaches the terminator region and newly created mRNA fall off along with the enzyme. This marks the end of prokaryotic transcription. The stages of transcription in eukaryotes are similar to that of prokaryotic transcription. However, there are some differences as well. Let's understand them in detail. Unlike prokaryotic transcription, which occurs in cytoplasm transcription, in eukaryotes, transcription occurs in the nucleus. There is only one RNA polymerase in prokaryotic transcription. However, eukaryotic transcription involves at least 3 RNA polymerases. The RNA polymerase 1 transcribes various rRNAs. On the other hand, RNA polymerase 2 transcribes the precursor of mRNA, known as heterogeneous nuclear RNA or hnRNA. RNA polymerase 3 is responsible for the transcription of tRNA, 5' subunit ribosomal RNA or 5SR RNA, and small nuclear RNA or snRNA. The processing of hnRNA marks another major difference between prokaryotic and eukaryotic transcription. The mRNA in prokaryotes does not require any processing. However, in eukaryotes, the hnRNA is inactive. Since it has a split gene arrangement with exons and introns, and hence it has to undergo processes such as capping, splicing, and tailing. The first process is capping, which involves an unusual nucleotide called methylguanosine triphosphate being added to the 5' end of the hnRNA. Capping helps mRNA to bind with small ribosomal subunits during protein synthesis. The next process is splicing, in which the introns are eliminated and exons are joined in a specific order. Finally, in the tailing process, about 200 to 300 adenylate residues are added to the 3' end of the hnRNA in a template-independent manner, giving rise to a poly-A tail. This process is also known as polyadenylation. After these processes, the hnRNA becomes mRNA and the transcription process ends. The mRNA can now be transported out of the nucleus and further used for the process of protein synthesis during translation. In 1962, James Watson and Francis Crick won the Nobel Prize for Medicine for deciphering the structure of DNA, the molecule that contains an organism's genetic blueprint. Through experiments, Watson and Crick had revealed that DNA was a double helix polynucleotide molecule, with each nucleotide consisting of the deoxyribose sugar, phosphoric acid, and nitrogenous bases adenine, guanine, cytosine, and thymine that are commonly called nucleotides. After the DNA structure was revealed, these scientists got busy finding an answer to the next question: how the nucleotides get converted into amino acids that synthesized proteins, the building blocks of life. These scientists already knew that DNA directs RNA synthesis, which in turn acts as a template in the synthesis of protein. They therefore suspected the presence of a genetic code, a set of rules in a gene that instructs a cell on how to synthesize a particular amino acid from the nucleotides of an RNA strand. The scientists now had only one aim: to decipher this genetic code. George Gamow, a Russian physicist, was the first to achieve some success in deciphering the code. He initially suggested that since the mRNA strand consists of only 4 types of nucleotides that code for the 20 amino acids in the human body, they need to combine to form a particular code. Through mathematical formulae, Gamow then proved that a permutation and combination of 3 nucleotides would be sufficient to make up a specific code for each of the 20 amino acids. The logic behind Gamow's postulation was easy to see. A single nucleotide, for instance, would code only 4 amino acids. Likewise, the combination and permutation of 2 nucleotides would result in codes for only 16 amino acids. However, the combination and permutation between 3 nucleotides would code for 64 amino acids, a little more than required. But nonetheless, Gamow stuck to his postulation. Though scientists were now aware that a combination of 3 nucleotides was all that was required to make a particular code, they had another question: which combination of nucleotides coded which amino acid? The answer to these questions came from research scientists Marshall Nirenberg and Heinrich Mattei. They created a free cell system by grinding E. coli bacteria using a mortar and pestle to obtain E. coli extracts. The E. coli was then added to a test tube containing everything needed for protein synthesis, including ribosomes and amino acids, to name a few. However, the test tube lacked an mRNA, without which protein synthesis was not possible. The scientists then added artificially synthesized mRNA consisting of only one base, uracil, in the free cell system. The E. coli extract in the free cell system was now able to produce a protein chain made of one repeating amino acid, phenylalanine, with the help of the Severo-Ochoa enzyme called polynucleotide phosphorylase that was present in the extract. This suggested triplet UUU codes for the amino acid phenylalanine. Nirenberg and Mattei had finally cracked the genetic code for at least one amino acid. Subsequently, Hargobind Khorana formulated a chemical method to produce well-defined nucleic acids, long strands of RNA with every nucleotide in the exact position. This helped determine the rest of the genetic code, which was expressed in the form of a chart. Today, we know that the genetic code is an array of 64 codons, a sequence of 3 nucleotides on an RNA strand that encodes a specific amino acid during protein synthesis. Of the 64 codons, 3 codons, UAA, also called ochre, UAG, also called amber, and UGA, also called opal, act as a stop codon, which means they terminate the translation process and as such don't code any amino acid. Hence, they are known as nonsense codons or termination codons. The rest of the 61 codons code the 20 amino acids found in the human body. Moreover, one codon codes for only one specific amino acid, which makes the genetic code unambiguous and specific. For example, the codon UGG codes for tryptophan, and AUG codes for methionine as well as acts as a start codon, which means it initiates the protein synthesis. The genetic code is also a degenerate, which means that many different codons encode a particular amino acid. Take the case of serine, which is coded by 6 different codons including UCU, UCC, UCA, UCG, AGU, and AGC. Phenylalanine, on the other hand, is coded by codons UUU and UUC. Did you know that the genetic code is almost universal and the same throughout all living organisms? The codon GUG, for instance, codes for amino acid valine in plants as well as animals.